We'll be delighted to have him, of course. Good morning, everyone, and welcome to International Academy of Cardiovascular Science webinar series. I'm AJ Marion, along with my co-host, Dr. Devendra Agarwal, and our capable administrative assistant, Natalie Hernandez. We welcome you to the IACS webinar series. Thank you for joining us this morning. For some of you tonight, today is my distinct pleasure and honor to introduce our webinar speaker, Stephen E. Nissen, MD, MACC. Dr. Nissen is an eminent physician scientist in cardiovascular medicine. He is the Chief Academic Officer for the Heart and Vascular Institute at Cleveland Clinic. He's an endowed chair in cardiovascular medicine and professor of medicine at School of Medicine at Cleveland Clinic. And Dr. Nissen has been the chair of numerous study sections and is a unique, really, imprint in the course of cardiovascular medicine, which are really indelible marks that change the care of patients with cardiovascular medicine. Indeed, rare are individuals who have made such an impact in the care of patients with cardiovascular disease. Briefly, Dr. Nissen received his MD from University of Michigan, and this was followed by training in internal medicine at UC Davis, cardiology fellowship training at University of Kentucky, and followed by joining the faculty there until 1992, when he was recruited to lead the clinical program, including intensive care, coronary care unit at Cleveland Clinic. And he soon became the chairman of the Department of Medicine in 2006, the position that he held for over a dozen years, and was appointed as chief academic officer in 2019, is the position that he currently holds. Dr. Nissen initial research, which was my first introduction to his impactful discoveries, was the use of intravascular ultrasound, IVUS, for characterization of coronary plaques and assessment of progression or regression of coronary atherosclerosis and response to therapy. He published over a dozen impactful articles in New England Journal of Medicine and JAMA, which truly established IVUS as the method, probably the gold standard to go to assess coronary atherosclerosis, and it really illuminated how rudimentary and crude coronary angiography is in assessing severity and extent and progression or regression of atherosclerosis. And his foray into clinical trial was a landmark study in 2007, which he and his colleagues showed rosiglitazone, which was a commonly used diabetic medication, increased the risk of cardiovascular disease, particularly myocardial infarction, which he published in 2007 in New England Journal of Medicine. This is an article that has received over 6,400 citations and of the most cited articles in cardiovascular medicine. Dr. Nissen followed that by conducting a number of clinical trials, serving as the chair of this clinical trials for numerous studies, which we will hear, including lipoprotein a, which I will not go into that, so we will listen to his presentation today. One of the characteristics of Steve, which is also shown in his introductory slide, is that his service is pro bono, and very few people are like Steve. Because he's heavily involved in industry, all of his activities and honorarium goes to charity, and which is quite an honorable thing to do. Dr. Nissen has published its scientific contribution in over 700 original research articles, including 200 articles in New England Journal of Medicine, JAMA, and Circulation alone. He has been cited over 100,000 times. That brings him an H index of close to 130 per Google Scholar. He is considered one of the top 1% highly cited researcher per Clarivate since 2015, persistently top 1% of the highly cited investigators. Dr. Nissen has been recognized for his numerous scientific contribution by a number of awards. Notable among them is the recipient of Simon Dack Award for outstanding scholarship, which was given by American College of Cardiology, as well as Golden Achievement Award for Medicine. He is Time Magazine recognized Dr. Nissen as one of the world's top 100 most influential people in 2017. Dr. Nissen serves as in leadership position in numerous organizations. He is the past president of American College of Cardiology and is also chair of the FDA advisory on cardiovascular and renal drugs. We truly are delighted and honored to host Dr. Stephen Nissen, our speaker today at the International Academy of Cardiovascular Science webinar series. And I don't see Dr. Dan Rader yet, but we have a distinguished panelist. All are world-renowned experts in lipoprotein little a, and which collectively have made the lipoprotein a giant A, no longer little a. I see to my right Dr. Simikas. Dr. Simikas has a lifetime commitment to prevention or reversal of atherosclerosis and has worked extensively on LPA. He is a leading authority on RNA therapeutics. He is professor of medicine and director of vascular medicine at the University of California San Diego, and is the founding director of the vascular medicine program at UCSD. He has also a double appointment. He is the clinical development vice president for clinical development, Ionis Pharmaceutical. He is the cardiovascular franchise leader at the same company as well. Next to Dr. Simikas, I see Dr. Lawrence Sperling. Dr. Lawrence Sperling also has a lifetime commitment to prevention of coronary atherosclerosis and extensive work in preventive cardiology. He is the former president of American Society of Preventive Cardiology. He is Katz Professor in Preventive Cardiology at the University of Emory. He is Professor of Global Health in Rollins School of Public Health. Also at Emory University, he is the founder and former director of Emory Center for Heart Disease Prevention and Chief Medical Officer of Family Heart Foundation. He is the 2024 recipient of National Lipid Association Honorary Lifetime Achievement Award. Meanwhile, Dr. Dan Rader will join us anytime his schedule permits. He must have technical issues. As of yesterday, he was online to join us. Just to mention, he's the chair of Department of Genetics at University of Pennsylvania and has numerous accomplishments and is one of the world leading authorities on genetic lipid disorders. With that, I will turn the podium to Dr. Nissen. Again, I'm truly grateful to all of you, Dr. Nissen, Dr. Sperling, and Dr. Simiakis for joining us today to give the IACS webinar today. Thank you, Dr. Stephen.
AJ, Dr. Rader is here.
Okay, great. Well, thank you so much for that overly generous introduction. And it's challenging to give a lecture when the three panelists know at least as much or perhaps more than I do about the topic, but I will try to give this talk. I know there are some students and fellows in the audience, so I'll try to make this effective for a broad audience. Lipoprotein A is in fact emerging from darkness into the light, and particularly Dr. Simikas has been very much involved in making that happen. Now, it has been so under the radar that the New York Times wrote in a article in 2018, they called it a heart risk factor even doctors know little about. And as I will show you, that remains to be a major problem. So let's first talk about what are the levels that are risky, abnormal for patients. The thresholds for increased risk that have been reported in the literature are using two different units, and that's a really big problem that needs to be solved. There is an approximate conversion factor of about 2.4 to 2.5, but as several of you have written about, that's not a perfect approach. But risky levels are anywhere from around 125 nanomoles per liter on into 170 nanomoles per liter, and some people think risky levels are even lower. There is very good literature on the relationship between lipoprotein A and cardiovascular, atherosclerotic cardiovascular disease. If you look at the classical study from Copenhagen, levels above about 117 milligrams per deciliter were associated in their data with a really substantial increase in risk, but so were levels from 77 to 117, and even down to 30 milligrams per deciliter, there were statistically significant increased risks. Now, there's also been controversy about the relationship between LPA and the risk of stroke. And in fact, one of the major trials studying a therapy for lipoprotein A did not include stroke in their outcome assessment. But if you look at a meta-analysis, it's pretty clear that there is a relationship between lipoprotein A and the risk of ischemic stroke. There is no relationship between lipoprotein A and non-vascular outcomes. This is a pure atherosclerotic risk factor. Non-vascular death is not affected. So the question, of course, is how does lipoprotein A contribute to atherosclerosis? We could talk for an hour just about this topic, but let me just summarize very simply in this slide that I borrowed from the European Heart Journal. The particle contains ApoB, LDL cholesterol as a core particle, linked by a disulfide bond to ApoA. The core particle has oxidized phospholipid. It enhances foam cell formation and does all of the things that LDL cholesterol and ApoB do to promote atherosclerosis. The ApoA component is thought to be prothrombotic. It affects plasminogen activation, tissue factor, in clot permeability, platelet response, and all of the things that are prothrombotic. So here we have wrapped into a single particle, both an atherosclerotic particle and a prothrombotic particle, and we think of this disease as atherothrombotic. That is what atherosclerotic cardiovascular disease is, an atherothrombotic disease, and this particle promotes both components. Now, it turns out that the ApoA has analogy, it's structurally similar to plasminogen, and that, of course, plays a role in its prothrombotic tendencies. Now, let me tell you one of the big problems we are facing. I did a study called the Heritage Study, 48,000 patients with known ASCVD in almost 1,000 sites in 48 countries. And the most important thing we found is, unbelievably, only 13.9% of these patients had had an LPA level previously determined. That is why it has stayed under the radar, and that is what must change. Now, the distribution of levels in this ASCVD population is shown here in milligrams per deciliter. Some of these people were measured in milligrams per deciliter and some in nanomoles per liter. 20% of the population was above 70 milligrams per deciliter, and 13% were above 90 milligrams per deciliter. Most of us are in the blue. And I hope those of you on this webinar have had your lipoprotein A tested, and I hope you are in the blue region. But as you move to the right, you see that there's this long tail of people with high levels and high risk. If you look at this population in nanomoles per liter, some people were measured in nanomoles per liter. 26% of the ASCVD population had levels above 150 nanomoles per deciliter. Now, elevated lipoprotein A has a prevalence in the US and globally. This is from the work of Dr. Simikas, and I will tell you that if you look at a cut point of 60 milligrams per deciliter, there are 64 million people in the United States, 150 million in Europe, and 1.4 billion people on our planet with levels above 60 milligrams per deciliter. And if you'll go to the right, you see there are still substantial people, even if you just look at the top 10% of the global population. This is an equal opportunity risk factor. Men, women, older, younger, high body mass index, low body mass index, HDL levels, triglycerides, across the spectrum of other risk factors, lipoprotein A has a similar risk, regardless of who you are and what your other characteristics look like. Now, there's a lot of question about where does LPA come from. Most people believe that it originated in our ancient ancestors in sub-Saharan Africa, and these people migrated with the diaspora from Africa into Europe, South Asia, North America, Latin America, and you can see these patterns of migration for this gene as it spread around the world. Because it originated in sub-Saharan Africa, it should not surprise anyone that Black individuals have substantially higher levels than whites and Hispanics. And you see here levels in this study from the Dallas Heart Study that levels were about twice as high in Blacks as whites. It is important to understand that this is mostly a genetic risk factor. It is diet, lifestyle, the other things that we think about when we think about atherosclerotic cardiovascular disease have little or no effect on the levels of lipoprotein A and the risk associated with it. Now, it's important to understand that in this genetic disorder, both alleles contribute to lipoprotein A levels. So you see on the left an individual that has one allele that is coding for relatively high levels of lipoprotein A. The other allele, the second allele, relatively low levels, and the total concentration is the sum of both contributions. Needless to say, if you have two abnormal alleles, two alleles that are associated with high levels, you tend to have levels that are in the stratosphere and very high risk. The question, of course, is who should we screen? And I try to emphasize this with all of our fellows and colleagues that we must do cascade screening because of the genetic nature of this disorder. And here you see a very nice study relatively recently from JAMA Cardiology. If you will focus on the right panel, first-degree relatives have a 47% chance of also having a level above 115 nanomoles per liter. Secondary relatives, 32%. So if you think about this, siblings, children, parents of patients that we discover with a high lipoprotein A need to be tested. And that's a really big challenge that I hope everyone on this webinar will emphasize with their patients and colleagues. And in fact, family history seems to have an amplifying effect of the consequences of lipoprotein A. Here you see levels on the left in nanomoles per liter and milligrams per deciliter. If you have a family history of premature disease, the relative risk shown at the far right can be very high. If your level is in that top, few percent, and you have a family history, you have a fourfold increased risk of MI. And even if your level is just in the top 20%, your level is increased 2.5-fold. Very important to understand how family history interacts here. This is a pretty fascinating study recently conducted. This is a study of adult ASCVD based upon Finnish youth tested at age 9 to 24 years of age. Those that had an Lp(a) of greater than 30 milligrams per deciliter, that's not very high, had a very high, shown in gold at the left, a very high hazard ratio for developing ASCVD in mid-adulthood. It is a much stronger effect than other risk factors such as blood pressure and LDL and BMI, and it is even higher than those that smoked when they were 9 to 24 years of age. So clearly we need to know if young people have levels, and I recommend, and many now organizations are beginning to recommend, testing young people at a relatively young age, perhaps in their early 20s, to find those people who are going to be at risk later in life. Coronary calcium score also is an important amplifier of risk. If you have a coronary calcium score of greater than 100 and an LPA in the top quintile, the risk is almost 5-fold greater for developing ASCVD. And even levels of calcium that are relatively low, above 1, and a higher lipoprotein A confer an increased risk. LDL cholesterol and LPA interact in producing risk. And from this very nice study, you see that if you have a high LDL and a high lipoprotein A, this is a study in women, very large study, risk is as high as 4-fold. If you have just a high LDL or just a high lipoprotein A, risks are lower. And in this case, the cut point was again just 30 milligrams per deciliter for a level of risk associated with lipoprotein A. Now, there is some controversy about whether the risk from an elevated lipoprotein A is exponential or linear. The classical data from the emerging risk factor collaboration suggested a curvilinear pattern where there's not much risk at lower levels and then a gradual acceleration of risk as levels get higher. A much more contemporary study and a much larger one in the UK Biobank participants shows a relationship that looks pretty linear, very much like LDL cholesterol, down to relatively low levels measured in nanomoles per liter. We're talking about here, down to 50 nanomoles per liter, we still see this linear pattern. And so my view is that this is probably a risk factor that has a linear relationship across a very broad range. It is also important to understand that it is well established that it is a risk factor in primary prevention, not just in secondary prevention. Here you see that in those individuals that are in the top 10% for lipoprotein A, which was 216 nanomoles per liter, the 15-year risk of developing atherosclerotic cardiovascular disease shows a hazard ratio of almost twofold. So again, we need to find people before they're sick if we're going to identify those at risk and allow them, when new treatments become available, to be treated. Now, there is also a strong relationship between lipoprotein A and aortic stenosis. Here you see a recent study, really interesting, in 44,000 people in a Korean population. And what you see is that when lipoprotein A was greater than 100 milligrams per deciliter, the risk of aortic stenosis was increased almost twofold, and that risk accelerates over time, as you see in this 20-year follow-up of this very large Korean population. And we see this in Western populations as well. Clearly, this is a risk factor for aortic stenosis. It is also a risk factor for outcome without aortic valve replacement. So this is survival without aortic valve replacement. And in the top tertile, the hazard ratio here is about twofold. And this is a Western population. And finally, if you find moderate aortic stenosis in a patient with a high lipoprotein A, their progression rate is about 50% higher than it is for those that have lower levels. And again, here the cut point was around 60 milligrams per deciliter. So if you find moderate aortic stenosis and a high lipoprotein A, it's important to follow these patients more closely. One of the interesting issues is how atherogenic is lipoprotein A compared with LDL-C, and in this case with ApoB. And there's some pretty interesting data. If you look at genetic FH, the risk of 175 nanomoles per liter for lipoprotein A is quite similar to the risk of FH. Again, showing what a potent risk factor lipoprotein A is for disease when you get to these levels of around 175 nanomoles per liter. And in fact, if you look at the UK Biobank data, as Marston did, what you find is the hazard ratio for each 100 nanomoles per liter, about 40 milligrams per deciliter, for lipoprotein A was more substantial than the risk for similar levels of ApoB. So more atherogenic per particle than ApoB. Also important to understand that this disease is also prevalent and risky in younger women. If you take the top quartile of young women, these are premenopausal women, those with levels greater than 30 milligrams per deciliter have a long-term risk adjusted for all the other risk factors that's really quite substantial. And so again, gendered is not necessarily protect against the bad outcomes that we see with elevated lipoprotein A. It is also important for the interventional cardiologist to understand that doing a coronary intervention in a patient with a high lipoprotein A, these patients have a higher risk of in-stent restenosis or in-stent stenosis with levels greater than 50 milligrams per deciliter, the hazard ratio is about 1.5 if followed out for 10 years. So important, these patients need to be followed closely even after a coronary intervention. Now, what about treatment? Well, we wish that statins were helpful, and they are helpful at reducing LDL cholesterol, and they're absolutely indicated in these patients, but they actually slightly raise lipoprotein A. This is from meta-analysis, and I think most of us would agree that it's about a 10% increase in lipoprotein A with statin administration. But it is important to understand that in the Jupiter trial, those with LPA levels above the median and those with levels below the median had very similar benefits when treated with rosuvastatin. So do not allow the fact that lipoprotein A is elevated by statins prevent us from treating these patients with statins. Currently, apheresis is the only FDA-approved therapy for lipoprotein A lowering. There is some very, very weak data using historical controls that suggests there might be a favorable effect. This shows data on MI, PCI, and CABG, 2 years pre-apheresis and 2 years post-apheresis. It's highly confounded by the fact that apheresis also lowers LDL, and using historical controls, it's really not very good science. Not randomized, and an event obviously may have triggered apheresis. So lots and lots of caveats here. And it's time consuming, it's expensive at up to $4,000 a session, and it's really not very widely available. Another question that comes up is whether aspirin reduces risk of an elevated lipoprotein A. Well, there is some controversy, but the Women's Health Initiative sub-study reported that patients with a genetically confirmed SNP for elevated lipoprotein A, of course, had increased cardiovascular risk, and that risk was reduced substantially with the use of aspirin. And here is, from again, a study showing that there was, for the genotype shown here in this graphic, there was a clear, MACE benefit, but, not as much bleeding risk. However, the interaction p-value here is just barely significant. So I don't think the data are as easily interpretable about whether aspirin actually has a favorable effect or not. I do treat these patients with aspirin, but it's clear that if they have increased bleeding risk, I'm not so sure it's the best idea. What about PCSK9 inhibitors? I see patients, I have a clinic every Friday where I see essentially the entire day, I see patients with elevated lipoprotein A from all kinds of distances. And many of them are coming in on PCSK9 inhibitors. And I must tell you, the data are not very good. This is from the Fourier trial. And yes, there was a reduction in lipoprotein A, but I want you to look to the right in the gold bar. The highest quartile of lipoprotein A had only a 16% reduction in lipoprotein A with PCSK9 inhibitors, and that amounted to about 15 milligrams per deciliter or 36 nanomoles per liter for people with levels above 165 nanomoles per liter. In my view, that is not a lot of bang for the buck. Now, there is some data from the Fourier trial suggesting that there was a disproportionate benefit of PCSK9 inhibitors in individuals with LPA levels above the median. But note at the bottom that the interaction p-value was 0.7. It was not statistically significant in terms of the difference, and so it is inconclusive in my view, and the data are just not strong enough for me to be an advocate for using PCSK9 inhibitors in these patients unless they have another indication for a PCSK9 inhibitor. The good news is that help is on the way. There are 5 drugs in development and probably several more. I think there's several coming now out of China. You can see them here listed. Some are in phase 3. Some are still in phase 2. But there is also an oral drug I'll tell you about called muvalaplin. Let's walk through them. First of all, it's important to understand the structure of lipoprotein A. The rate-limiting step in synthesis of the LPA particles is apolipoprotein A. And if we can inhibit production of apolipoprotein A, we can in fact inhibit production of lipoprotein A. There are 3 approaches that have been used. Antisense oligonucleotides, pioneered by Dr. Samikas and his group at Ionis, shown here, single-stranded DNA, small interfering RNAs, which are double-stranded RNAs, and then a disruptor that blocks the association of ApoA and ApoB, and that's muvalaplin. I'll show you in a minute. Now, the term for these approaches is, for the injectable approaches, is RNA interference. An antisense oligonucleotide, what it does is the gene is transcribed, messenger RNA produced, and the antisense oligonucleotide degrades the messenger RNA for ApoA in hepatocytes and prevents the production of ApoA, or certainly dramatically reduces it, which prevents the formation of lipoprotein A particles. Now, one key development that was again pioneered during the development of the Ionis drug, now known as pelacarsen, was the use of a sugar known as GalNAc, or N-acetylgalactosamine, that has a strong affinity for a receptor in the liver and allows a approximately 2 log reduction in dose to achieve the same effect size on reducing lipoprotein A, an important breakthrough in development. This is now the phase 2 New England Journal of Medicine paper written by Dr. Samikas, showing that 20 milligrams given every week produced an 80% reduction in lipoprotein A, very clinically meaningful reduction. That was translated to a 80 milligram dose per month, which is how the drug pelacarsen is now being used in phase 3. And the phase 3 trial is ongoing at about 800 sites in over 40 countries, 8,000 patients with elevated levels. They're on optimized background therapy, planned duration of 4 years, and a 4-component MACE endpoint. The results are due in 2026. Some more recent approaches use a small interfering RNA approach, again using GalNAc conjugation. Intrahepatically, the double-stranded RNA breaks into an antisense strand, which is the warhead here, and it contains nucleotides chemically modified to resist degradation by ribonucleases. The first of these was olpasiran, developed by Amgen and now being studied in a phase 3 trial. And it produced in the larger therapeutic doses nearly a 100% reduction in lipoprotein A. The dose being used in phase 3 has not been revealed, but it hopefully is sufficient to produce high levels of reduction. Next to come along was zerlasiran, developed by a British company. I studied this and published the results in JAMA in 2024. And again, you can see that with 3 doses of 300 milligrams every 16 weeks, levels with some sawtoothing were reduced in the range of 80 to as much as 96%. The next drug to come along is lepodisiran. This is a somewhat unusual small interfering RNA that's a dicer substrate tetraloop siRNA. It contains GalNAc. It's longer and is actually eventually degraded into a more classical 22 nucleotide small interfering RNA. Whether the tetraloop structure or the dicer substrate has an important effect on results is uncertain. What I can tell you is that it did produce quite profound reductions in lipoprotein A. Here you see the study we published very recently in the New England Journal of Medicine, showing that a single 400 milligram dose actually produced reductions that were clinically meaningful out to 540 days. And similarly, 2 injections, 180 days apart, produced over a 90% reduction for a full year. Here is the key primary endpoint from that New England Journal of Medicine publication. From days 60 to 180, and 94% reduction. And when given in 2 injections, 95% reduction on time averaged all the way out to a full year. The oral drug, muvalaplin, is also in development, soon to enter phase 3. And again, the way it works is it disrupts the association between ApoA and ApoB, preventing formation of lipoprotein A particles. This paper was written by my former postdoc, Dr. Steve Nichols. And what you see is in the phase 2 trial, doses of anywhere from 60 to 240 milligrams produced an 80 to 85% reduction in lipoprotein A. This is given daily, orally, out to week 12, and appears to have a substantial reduction that is at least durable out to 12 weeks. A big controversy is how much lipoprotein A reduction is required to reduce cardiovascular events. And here you see several manuscripts that try to translate Mendelian randomization studies along with clinical trial estimations. And what you see is that it appears that anywhere between 50 and 100 milligrams per deciliter would be sufficient to produce a reduction in MACE similar to a 1 millimole reduction in LDL cholesterol. Those are reductions that are being achieved in the phase 3 trials. We will find out relatively soon whether these predictions are in fact accurate or not. Keep in mind that in the Mendelian randomization studies, we're talking about lifelong exposure. We are only talking about exposure for 4 to 5 years in clinical trials, so that's why these are somewhat different. So let me just tell you that there are drugs in development that have a profound effect on lipoprotein A, ranging from about 80 to 100%, and an oral drug that is in development. We are now entering a new phase. And the phase in the study of lipoprotein A now takes us to the phase 3 trials of emerging therapies. And they are designed to determine if reduction of LPA can achieve clinically meaningful cardiovascular risk reductions. These trials have enormous public health consequences. I have an appeal to all of you. We are very hopeful that we will be successful in the first of these trials, which is the pelacarsen trial. If we are successful, we need to know who to treat. That means that ASCVD patients need to be tested, people with family histories need to be tested, and frankly, I think that young people need to be tested. If we don't know who to treat, when these drugs succeed, and I think they will succeed, we will remain in the darkness. And the New York Times article said that this was a heart risk factor even doctors know little about, will persist. So we need to test more people. Thank you for your attention. I'm looking forward to the panel discussion, and I left about 15 minutes for the discussion. Thank you very much for your attention.
Thank you, Dr. Nissen. Thank you for your really in-depth, comprehensive, insightful, fabulous presentation. This is a fantastic webinar. We truly are grateful to you. With that, I'd like to acknowledge Dr. Daniel Rader, who joined while I'm making introductory comments. Dr. Daniel does not really need introduction. He's a world-renowned authority on genetics of lipid disorder. He's a chair of the Department of Genetics at UPenn. He is an endowed professor in molecular medicine, chief of translational medicine, director of preventive cardiology, and scientific director of clinical and translational research program at Cardiovascular Institute of Penn. He is also deputy editor of ATVB and a member of National Academy of Medicine. Dr. Rader, with that, I'm going to ask you the first question. Or would you like to ask the first question, rather? Well, we heard a lot about genetics of it. And, your group has pioneered base editing. Is there any room for base editing for LP(a) or LPA gene?
I think so. I think this is a protein, ApoA, that has no known important physiologic role, at least currently. And there are many people who have essentially undetectable levels of LPA who don't seem to be at any increased risk for anything else. So I think just like with PCSK9, we're not only inhibiting it or silencing it, but now essentially knocking it out using CRISPR gene editing. Is being done for PCSK9. I think that same approach could be and is being pursued for LPA. And I do think that in addition to all the other approaches that Dr. Nissen discussed, I think that will be another approach that will be pursued over the next several years.
And just to comment that, in fact, we are involved in just such a program and moving along. And it's a little bit longer timeline to get to the approval stage, but I'm very excited about the possibility of curing this disorder with gene editing.
Very good, thank you. Comments from our panelists, Dr. Sperling, Dr. Simikas. You all have worked extensively on LP(a). Your thoughts?
Sam, do you want to start off? I have a lot to say, but I know you do as well. We have the LP(a) team here.
Feel free to go ahead. I'll follow up.
Okay, great. So first of all, Dr. Nissen, that was one of the best overviews I've ever heard regarding LP(a). So thank you for that. Maybe as a take home, I'm going to talk about the 3 A's related to LP(a). LP(a) clearly has arrived after 6 decades of scientific investigation, moving to translation to pharmacotherapies. LP(a), the story that you heard today, is honestly one of the most exciting and potentially impactful areas in all of medicine. So look forward to the RCTs and their outcomes. So the arrival of LP(a), LP(a) is here, but in order to, as Dr. Nissen said, this is a public health concern. 1.4 billion people on the planet potentially are living with a high LP(a). So we must be aware. And less than 1% of the general population has had their LP(a) measured. the Family Heart Foundation, and other such organizations have really promoted awareness of Lp(a). In fact, universal Lp(a) testing is recommended in Canada and Europe. We're awaiting the updated guidelines in the US here. And then the last one is just ASCVD and AS risk reduction. And often with Lp(a), we are countered with why measure it if there's nothing we can do about it. And there's a lot we can do. First of all, just awareness alone, cascade screening. But beyond that, we know that cardiovascular risk reduction strategies do favorably impact those living with an elevated Lp(a), so lowering LDL, and even lifestyle behavioral approaches. We published a paper in the European Journal of Preventive Cardiology. If you adhere to Life's Simple 7, now Life's Essential 8, by following a healthy lifestyle behavioral approach, you can reduce the consequences of elevated Lp(a). That continuous risk, though, linear risk, is something to pay attention to. And I know Dr. Samikas has dedicated his career, and we all learn from him and Dr. Rader, but probably all living with Lp(a) are not the same. And those with the very high Lp(a) levels, established ASCVD, and recurrent events hopefully will be first in line for these critically important therapeutic agents. So, Sam, so that's the 3 A's, my summary.
Great. Well, thanks. Well, let me just say that, Steve, thank you very much for that. A really outstanding presentation that summarized the field beautifully. You know, there was a point when nobody cared about LPA, and despite the fact that there's not as much testing as we'd like, it's really, I think, turned the corner. I think it's now on a lot of people's radar, and a lot of people are talking about it. So it's been fantastic. And we see this from the fact that there are over 10 drugs now in development, which is an amazing thing for the field. I thought I would just maybe two things that maybe we can discuss, Steve. One is, what do you think could go wrong in the trials? And from the perspective, as you know, Dan mentioned this, but we really don't know if there's a physiological function. Could something show up in a pharmacological study that hasn't been shown up in epidemiology? And specifically, I'm speaking to, you know, we know there's an association with diabetes at low levels. Yeah. The other part of that is, you know, if the LDL is so low, will patients get a benefit? Yeah. So I wonder if you could address that. And I'll just ask my second question real quick, which is the oral agent, I think, shouldn't be considered in the same realm as the other drugs because it doesn't actually lower LPA by itself. It allows for clearance mechanisms. And I'm worried that some of that clearance could be in the vessel wall. And I'm wondering if you might address that also as a second part of that question.
Well, those are just great questions. And listen, I lose sleep at night worrying about our trial, Sam. And you know, we did something which I think was the correct and ethical thing to do, but you know, we have to overcome it. We mandated that everybody in the LPA Horizon trial studying pelacarsen be treated very intensively for all their other risk factors. And they will have low LDLs, and they will have well-controlled blood pressure, and all their other risk factors well-controlled. And so we're going to have to show that lowering lipoprotein A on top of very good background therapy will produce a clinically meaningful benefit. So that's one thing I worry about. I worry less about some surprise like a big increase in diabetes. And, you know, partly because we have a very good data monitoring committee that's monitoring the trial. And if there were concerns, they would have probably said to us, please, take care to look carefully and treat any increases in blood sugar in patients. We haven't heard anything. And so I think the safety side doesn't worry me. The efficacy side does, keeping in mind that in the Mendelian randomization studies, it's a lifelong exposure. And we're trying to do something in 4 or 5 years that will produce a clinically meaningful benefit. And that is another risk for these trials. So I'm optimistic, but I'm also doing a little hand-wringing. Now, the muvalaplin story, I completely agree with you. We don't know what happens when you have all that free ApoA floating around, and it's why it must be studied in an outcome trial before it can be approved. And that will be done. And I think we will, wait and see. And I think that it's going to be a while before the FDA is going to say anything that lowers lipoprotein A is approvable, which is kind of what they've done with LDL. So that's just my view.
Yeah. Just add a little nuance on the drugs do have different potencies. I suspect some of the non-pelacarsen drugs, patients are going to be getting a lot of zeros on their LPA.
Yes, they will.
And with pelacarsen, we had a 120 milligram dose that lowered LPA 92%, but we chose not to use that. And so what you might see in the trials is that there's going to be a lot more zeros for the other drugs. And I don't know if that's going to make a difference or not, but that's kind of an interesting area where they'll have to follow.
Yeah, we will have to. In fact, every drug will need, for now, every drug will need to prove itself in a clinical outcome trial. And the trials are getting bigger and bigger.
Yes.
Fantastic. So we have one question from audience. Dr. Mendel Chan, who has done seminal work on small dense LDL. He has a question here. Is free ApoA innocent? If so, is breaking the disulfide bond more important than reducing ApoA production, as shown in the oral drug trial you mentioned? Does ApoA rise acutely at ST elevation, myocardial infarction, when LDL-C is usually low? If so, why? Because if LPA is genetically determined, why would it rise acutely at ST EMI if so? Question by Dr. Mendel Chan.
Yeah, I can't answer that, but maybe one of our panelists, Dan or Sam, one of you guys can help answer that question.
I can take a stab at the first part. I think, about 20 years ago, we tried to disrupt the LPA particle using mipomersen, which is an inhibitor of ApoB. We did this in transgenic mice. It did lower LPA as you measure it in the whole particle, but we noticed that the liver kept making free ApoA, and that free ApoA had the OXPL on it. And we got very worried that that wasn't really an ideal way. So we went back to the drawing board and developed an inhibitor to ApoA. So when you inhibit the particle assembly, what you're hoping is that the ApoA now clears. So what are the mechanisms of that clearance? There's a variety of receptors in the liver cells, LDL receptor, for example, plasminogen receptors, VLDL receptors. There's clearance through the renal pathway, but it also could accumulate in tissues and vascular tissue. So as was mentioned, we need to really know the safety of that. It would be good for the company to try to do a study to track it, where it goes once they disrupt the assembly. I think in a preclinical model, that would be useful information to have. The second part of the question was, I think, unrelated, but that was on an ACS. LPA has an IL-6 response element. So if you have an inflammatory stimulus, particularly if it's large and acute, you will have an increase in LPA during STEMI, and it takes 3 to 4 months for it to become quiescent, which is another little quirk of this whole story. I don't think it's going to impact us that much, though, because the patients have high levels will be high when they come in. And so it may be an issue when people have borderline levels. We may have to check them twice. Dan?
Yeah. Sam, I just want to add, Sam, in our studies, we're not studying people immediately after MI.
Yeah, great point.
Thank you. Now, concluding remarks from panelists and our distinguished speaker. Anyone wishes to make any concluding remarks? It's time for it.
Dan?
Then the plot shows Lp(a) has the largest effect size in terms of risk of MI. What do you say? Is there any monogenic familial form of LPA mutations?
Is that question for me? So clearly the genetics are the major dominant factor that play a role in LPA levels. If the question is, are there, quote, monogenic forms of high LPA? I guess the answer is yes, because it's basically the ApoA gene or LPA gene that actually determines the levels of the LPA. But it's complicated, and it's not just due to one variant, and it's due to repeats of the Kringles and so forth. But yes, the LPA gene is the gene that largely determines LPA levels. I will say, though, that there are other genes that contribute to LPA levels, but in a much more modest way. Ali, I know we're summary. I just want to say this, Steve mentioned this. This is one of the great experiments in cardiovascular medicine over the next several years, and it has tremendous public health significance, as Dr. Nissen mentioned, but also just tremendous biological and medical interest in terms of how this plays out. So I think we all are on the edge of our seats waiting. And Dr. Nissen focused on coronary disease, and clearly coronary events are the endpoints for these trials. But the fact that LPA is the strongest genetic and epidemiologic risk factor for aortic stenosis presents yet a whole nother opportunity for a new medical therapy for aortic stenosis, for which we have no medical therapies. And I think it's just one other aspect of this whole story that will unfold over the next decade. So very, very exciting for both coronary disease and for aortic stenosis.
Fantastic. Thank you. Any other comments before we ask Dr. Agarwal to make concluding comments on behalf of IACS?
Just adding, clearly a causative risk factor. This magnifies risk, as Dr. Rader said. This is a tremendous opportunity for impact, and this is an area that we should buckle our seatbelts. Clearly, over the next 5 years, we're going to learn a lot, and look forward to be part of that learning and part of the community related to awareness of Lp(a) levels.
Thank you, Dr. Sperling. Dr. Agarwal, would you make a couple of words about IACS webinar series, IACS, and to conclude the session?
Yes, sir. Thank you. Once again, thank you very much, Professor Nissen, for a wonderful talk. And I also really thank all the other panel members for their comments and suggestions. Such webinars are very useful for our young generation as well as for older generation as well, but especially the young medical school students who are attending these webinars. Our goal of the Academy of Cardiovascular Sciences is to promote the cardiovascular research and mentor and train young generation. So this is one of the initiative under the leadership of Professor Marian. And we all really thank you for your support, and also request you to please join the Academy of Cardiovascular Sciences and encourage your fellows and residents to join the Academy and attend the annual meeting. Thank you very much.
Thank you all. Thank you, Dr. Nissen. Thank you, Dr. Sperling, Dr. Simicas, and Dr. Rader. Grateful to you all. And thank you, Natalie, for organizing this. Without you, it would not have been possible. And thank you, audience. Thank you, everyone that attended. And we're looking forward to having this kind of impactful webinars that are truly going to change the course of medicine. Thank you, Dr. Nissen. Grateful to you all, and have a wonderful day, all of you.
Thank you for having me.
Absolutely a pleasure and honor. Thank you, Steve. Bye-bye.
Thank you, AJ, for your leadership.
Thank you.
Thank you, Natalie.
Thank you. Have a safe trip.
Yeah, thank you. Yeah. Okay, bye for now.